Identify the correct match from the following matching: physiological effect and hormone, fruit ripening, flowering, stem and root elongation, senescence, tropism causing growth, stress hormone, gaseous hormone, genetic dwarfism removing hormone. A3, fruit ripening, gaseous hormone, that is ethylene. Flowering is genetic dwarfism removing hormone, that is gibberellins. Flowering in poinsettias, flowering in dianthus and other plants. Stem and root elongation, tropism causing hormone. Stem and root elongation, tropism causing hormone means indole 3-acetic acid or auxin. Senescence, premature leaf and fruit fall is due to stress hormone, that is abscisic acid. So fruit ripening, gaseous hormone. Flowering, genetic dwarfism removing hormone, gibberellin. Stem and root elongation, tropisms causing hormone, that is auxins. And senescence, stress hormone, that is abscisic acid. Identify the untrue statement from the following, that is false statement. The most abundant as well as actively natural auxin is IAA, correct. Indole 3-acetic acid. Shoot and root apical meristems are the site of biosynthesis of auxins is correct. The ratio between N atoms and carboxyl group in IAA molecule is 1:1 is correct. So untrue statement is second one. Auxins move in large quantities towards the apex than towards the base. Basipital movement is more in auxins than in acrobital movement. So auxins move in large quantities towards the apex than towards the base is wrong. So second option, auxins moves in the large quantities towards the apex than towards the base is wrong. The most abundant as well as active natural auxin is IAA, correct. Shoot and root apical meristems are the sites of biosynthesis of auxins is correct. The ratio between nitrogen atoms and carboxyl groups in IAA molecule is 1:1 is correct. So wrong is, untrue is second statement. This is again a matching. Study the following list and identify the correct match. Phenyl acetic acid, indole butyric acid, 2,4,5-trichlorophenox acetic acid, naphthalene acetic acid. Naphthalene acetic acid, phenyl acetic acid, trifluorophenox acetic acid, indole butyric acid and 2,4,5-trichlorophenox acetic acid. PIA is phenyl acetic acid second. IBI is indole butyric acid 4. 2,4,5-T is 2,4,5-trichlorophenox acetic acid 5. Na is naphthalene acetic acid 1. So A2, B4, C5, D1. These are the expansions of this particular. PIA is a natural auxin along with IAA. IAA, 4-chloroindole acid and phenyl acetic acid are natural auxins present in plants. Synthetic auxins are indole butyric acid, naphthalene acetic acid, 2,4-D and 2,4,5-T. So first option for this. Next, study the following list and identify the scientists. Power of movements in plants. Tropic movements are induced by auxins. Accumulation of high concentration of auxins in axillary buds than apical buds. Organ forming substances are produced in leaves. A1. The power of movement in plants by studying the tropic movements in canary grass plants is written by Charles Darwin. Tropic movements are influenced by auxins, Kalodny and Wendt theory. The amount of auxins at the stem apex will be less, whereas in the root tip are more. That's why root always show positive zeotropism and stem always show positive phototropism was proved by Kalodny and Wendt. Accumulation of high concentration of auxins in the axillary buds than in the apical bud. So all the auxins formed in the apical bud move downwards, settles in the axillary buds and stops the growth of axillary buds is called apical dominance, which was proposed by Thiman and Skoog. Then organ forming substances are produced in the leaves. Organ forming substances are produced in the leaves were proposed by Sachs, J.V. Sachs. So answer is A1, B2, C3, D4. C3, D4. Arrange the following phytohormones in descending order according to their number of carbon atoms in each molecule. GA20 is 20 carbon, ABA is a sesquiterpene 15 carbon, indole acetic acid, GA1 is 20, ethylene is C2H4, that is 2 carbon. So B, GA20, so it is in an descending order, top to bottom, GA20, next D, BDACE, BDACE, first GA20, then indole acetic acid, then GA1, 20 and 19, then ABA, 15, then indole acetic acid, finally ethylene. BDACA is based on the carbon atoms present in them. Phytohormone that can be synthesized from the connecting link in aerobic respiration. What is the connecting link in aerobic respiration? Acetyl coenzyme A. So acetyl coenzyme A is the precursor compound to produce. What are the hormones? One, gibberellins, other abscisic acid. Abscisic acid is normally formed by catabolism and anabolism. Anabolism is from acetyl coenzyme A, catabolism by mevalonate pathway where carotenoids are broken. Mevalonate pathway where carotenoids are broken is catabolism by which ABA is produced. Anabolic process is from acetyl coenzyme A. So gibberellins and abscisic acid both are produced from acetyl coenzyme A. So phytohormones that can be synthesized from connecting link in aerobic respiration. Connecting link in aerobic respiration is acetyl coenzyme A. It is a connecting link between glycolysis and Krebs cycle. So gibberellins and abscisic acid are the two phytohormones obtained from acetyl coenzyme A. So answer both A and 1 and 2. Assertion. ABA is called stress hormone. Induces stomatal closure in drought conditions. Whenever there is a water loss or stress condition, ABA concentration increases and remove all the potassium ions present in the guard cells. Whenever potassium ions are moved out of the guard cells, chloride ions moves out, malic acid comes out, protons again enters the guard cells, which is explained in stomatal mechanism. By all these conditions, flaccidity of the guard cells increases and stomata closes. So ABA is a stress hormone. It induces stomatal closure in drought condition. So because ABA is a stress hormone, it induces stomatal closure in drought conditions. So A and R are correct. R is the correct explanation of A, first option. Study the following table. Precursor, site of synthesis, physiological effect. Carotenoids, dormant parts, closure of stomata. Carotenoids, senescent leaves, opening of stomata. Tryptophan, root and stem apex, diotropisms. Methionine, ripening fruits, wound healing. Select the correct pair of answers in which farmer is in the pair shows set of characters related to ABA and later is the pair shows set of characters unrelated to IAA. Related to ABA, unrelated to IAA. Answer is 1 and 4. Carotenoids, dormant parts, closure of stomata are related to ABA, abscisic acid. They are formed from carotenoids, mevalonate pathway. They are present in dormant parts and closure of stomata. Then set of characters unrelated to IAA. Unrelated to IAA is methionine, ripening fruits, wound healing. They are not related to indole 3 acetic acid. So 1 and 4 is the answer for this. A and B are two phytohormones. A is useful in increasing shelf life of lettuce and spinach. A and B are two phytohormones. A is useful in increasing the shelf life, that means freshness of lettuce and spinach. B promotes synchronous senescence of Nicotiana leaves. B promotes synchronous senescence in Nicotiana leaves. Which one of the following pairs the former represents A and latter represents B respectively? A and B are two phytohormones. A is useful in shelf-life period of lettuce and spinach is cytokinins. This is called Richmond and Lang effect, where leafy vegetables remains fresh for many years by the treatment of cytokinins. B promotes synchronous senescence of Nicotiana leaves. Nicotiana leaves are useful only after becoming senescence. The senescence of Nicotiana leaves is increased by ethylene. And another important feature is ethylene also reduces the percentage of nicotine, dangerous nicotine in Nicotiana tobacum leaves. So answer for this, A and B are two phytohormones. A is useful in increasing the shelf life period of lettuce and spinach. B promotes the synchronous senescence of Nicotiana leaves. The A and B respectively cytokinins and ethylene. It is not gibberellins and ABA. It is not auxins and gibberellins. It is not zeatin and NEA. It is cytokinins and ethylenes. Ethylene inhibits. Ethylene, what is the function of ethylene? It inhibits stem elongation. It inhibits stem elongation is correct. Stimulates stem thickening. thickening of the stem is not stimulated by the ethylene. It causes horizontal growth. It causes horizontal growth. These two properties belongs to triple response growth of ethylene. So ethylene inhibits stem elongation is correct. Ethylene causes horizontal growth is correct. Ethylene is not at all related in stimulating stem thickening is wrong. So answer is 1 and 3, third option. Senescence is promoted by. Senescence is promoted by. Senescence is always promoted by ethylene and ABA. Cytokinins always delay the senescence. What is senescence? Destruction of chlorophyll in the leaves, making the yellowing of leaves and making the leaves to fall. So whenever delay the senescence, the leaf remains fresh for many days. Leaves remain fresh for many days. This property is popularly called Richmond and Lang effect. Richmond and Lang effect. This is the property shown by cytokinin. Whenever cytokinins like BA, benzyl adenine, BAP, benzyl aminopurine, are added to the leaves, definitely the senescence of leaves is delayed and leaves remain fresh for many days. This is called Richmond and Lang effect. So senescence is not promoted by cytokinin, it is promoted by ethylene and ABA. So answer is 2 and 3. These are VD sites. Just now we discovered. We discussed about this 2,4-D and 2,4,5-T. 2,4-D is dichlorophenoxyactic acid, 2,4,5-T is trichlorophenoxyactic acid, 6-furfuryl aminopurine is the cytokinin kinetin extracted by herring sperm DNA by Miller and Skoog. So 6-furfuryl aminopurine is a cytokinin called kinetin obtained from the herring sperm DNA. So 2,4-D, 2,4,5-T are the two very popularly known VD sites. Is a 3-column tabular form. Phytohormone, precursor of for its synthesis and main influence. Indole 3 acetic acid, tryptophan, cell elongation. So indole 3 acetic acid auxin is always formed from tryptophan and it helps in cell elongation, correct. GA3 acetyl coenzyme A, cell division. Gibberellic acid 3 has come from acetyl coenzyme A, but main influence is not cell division, internodal elongation. So it is wrong. ABA acetyl coenzyme A, dormancy. Abscisic acid is formed from acetyl coenzyme A. It increases dormancy, that is rest period in the case of almost all buds and seeds. Ethylene, cystine, fruit ripening. Ethylene fruit ripening is correct, but not come from cystine. It comes from methionine. So IAA, tryptophan, cell elongation is correct. GA3 acetyl coenzyme A, cell division is not correct. ABA acetyl coenzyme A, dormancy is correct because they cause dormancy. And ethylene, cystine, fruit ripening. Cystine is wrong. It is methionine. So answer is 1 and 3. Next, another 3-column tabular form. Phytohormone, type of phytohormone and main feature. IAA, growth promoter, male flower formation, wrong. IAA is growth promoter, correct, but it helps in the formation of female flowers in cucurbits, not male flowers in cucurbits. Zeatin, growth inhibitor, Richmond and Lang effect. Zeatin is the most naturally occurring cytokinin obtained from the endosperm of maize by Miller and Latham independently. Miller and Latham extracted this zeatin from the endosperm of Zea mays. It is not a growth inhibitor, it is a growth promoter. So it is wrong. Growth inhibitor is wrong, but Richmond and Lang effect is correct. GA3 growth promoter stem elongation. GA3, growth promoter stem elongation. ABA, growth inhibitor seed dormancy. So IAA, growth promoter male flower formation is wrong. Giatin, growth inhibitor is wrong. GA3, growth promoter stem elongation is correct. GA3, growth promoter stem elongation is correct. ABA, growth inhibitor seed dormancy is correct. So the wrong answers are 1 and 2. The correct combinations are 3 and 4. Correct combinations are 3 and 4. So second option. This is another 3-column tabular form. Phytohormone, crop in which it is used, purpose. Phytohormone, crop in which it is used and what is the purpose of using it. 2,4-dichlorophenoxyastic acid used in rice crops to eradicate dicot weeds is correct. Completely eradicate dicot weeds is correct. Kinetin, spinach to increase shelf life period of leaves. Kinetin, Ka, is nothing but cytokinin. Definitely added to spinach leaves and to increase shelf life period. Richmond and Lang effect is correct. ABA, potato, to increase potato production is wrong because they stop the sprouting in potato. They always definitely stop the sprouting in potato, so they decrease the potato production. So ABA, potato is correct, but to increase potato production is wrong. Ethylene, tobacco, Richmond and Lang effect is wrong. Ethylene, when added to tobacco, senescence is increased. Richmond and Lang effect is decreasing senescence or minimizing senescence or stopping senescence. So ethylene is added to tobacco, correct, but not for Richmond and Lang effect, but to increase senescence and decreasing the nicotine content. So answer 2,4-D. Crop in which it is used is rice to eradicate dicot weeds is correct. Kinetin, spinach leaves to increase the shelf life period of leaves is correct. ABA, to potato to increase potato production is wrong. Ethylene, to tobacco, Richmond and Lang effect, delaying in senescence is wrong. Increasing senescence is correct. So the two correct combinations are 1 and 2. Arrange the following events in order that explains the seed germination in barley. How seed germination occurs in barley? First, release of zibberellins from embryo. Always zibberellins are released from embryo. Then these zibberellins which are released from embryo directly go to the alluron layer, a triploid proteinaceous layer present around the endosperm or in between endosperm and embryo in monocot seed. So synthesis of alpha amylase or alpha amylase enzyme is done by zibberellin by going to that place. What is that place? Alluron layer. Then hydrolysis of starch to glucose. Whenever amylase is formed in the presence of water, amylase is a hydrolytic enzyme, converts the complex starch into simple glucose. Then utilization of transferred sugars by embryo. Utilization of transferred sugars by embryo to break the seed coat. So first is release of zibberellins from embryo, correct. Then synthesis of amylase in alluron layer after zibberellin transporting nearer to the alluron layer. Third, hydrolysis of starch to glucose by amylase. Hydrolysis of starch to glucose by amylase. Utilization of transferred sugars by embryo. So DABC is the correct sequence in this. Now we will discuss the bits from crop improvement. Pure line selection is more an art than a science. Pure line selection is more an art than a science, more a science than an art, both art and science, only an art. It is more an art than a science, mass selection and pure line selection. Select the right features related to emasculation. Right features related to emasculation. So total hybridization concept is studied in various steps. One is selection of parents. 2, selfing in parents. Selfing in parents means even cross-pollinated crops are made to perform self-pollination, which is called inbreeding. Emasculation, removal of stamens from the bisexual flowers of female parental plant. The covering of emasculating flowers by polythene covers is bagging. Emasculation is done to avoid unwanted self-pollination. Bagging is done to avoid unwanted cross-pollination. Artificial cross-pollination, pollen grains are brought from the male flowers and deposited on the female flowers of female flower by removing bags. Again bagging is done. F1 generation is formed. Seeds are sown to produce F2 generation. These are various steps in hybridization. In this, these two are very important. Bagging is done before pollination and after pollination. Bagging is always done before pollination and after pollination. So, select the right features related to emasculation. It is done in male parent is wrong. It is always done in female parent in the flowers before anthesis. Before anthesis means before opening of flowers. It is done both in male and female parent is wrong. It prevents unwanted cross pollination. Unwanted cross pollination is wrong. It prevents unwanted self pollination. Unwanted cross pollination is prevented by bagging. So, it is done in male parent is wrong. It is done in female parent is correct. It prevents self pollination is correct. So, 2 and 4 are correct. Emasculation is not necessarily for. Emasculation is not necessary for self pollinated crops wrong. Male sterile lines. Male sterile plants are produced in Brassica napus by genetic engineering transgenic methods. Male sterile plant means it is not producing pollen grains, even stamens are present. Even though stamens are present, those stamens cannot produce pollen grain. They are called as male sterile plants. Then polygamous plants. Polygamous plant means separate male flower, separate female flower, separate bisexual flowers are present on the same plant. Here emasculation must be done. Then dioecious crop. Dioecious crop means separate male plant and female plant like Areca boraces. In these flowers show either androecium or gynoecium. If both androecium and gynoecium are there, then only emasculation is required. If dioecious plants with separate male and female flowers are there, there is no question or no chance of emasculation. So, self-pollinated crops is wrong. Male sterile plants is correct. Polygamous plants is wrong. Dioecious crops is correct. So, emasculation is not necessary for male sterile plants and dioecious crops. 2 and 4. Second option. Identify the sequence of development of various plant breeding methods by man. Various plant breeding methods by man means time after time. First is selection. First is always selection. Then is introduction. Even for selection, introduction also plants has to be selected. What is introduction? Transferring the plants from their normal locality to a completely new locality with entirely different climatic conditions is called as introduction. Even though introduction is said to be the first and foremost method in crop improvement, even in introduction also in that area where the plant is growing, they are selected whether they are good plants or not. So, selection is the first method. Introduction is the next method. Next is hybridization. A cross made between two plants with different genotypes is called as hybridization. New varieties are produced. Then a little bit advanced method is mutation breeding. More than mutation breeding is polyploidy breeding. So, identify the sequence of development of various plant breeding methods by man is first is selection. First is always selection either by nature, natural selection or by human being, artificial selection. If cross-pollinated crops are given importance, mass selection. If self-pollinated crops are given importance, pure line selection. Pure line is, what is pure line? The progeny of a single self-fertilized homozygous plant is called pure line. The statement is correct, important. The progeny of a single self-fertilized homozygous plant is pure line. So, first option, first selection, then introduction, then hybridization, finally mutation breeding. These are the development of various plant breeding methods by man, one after the other in a sequence. So, first option. Arrange the following stages of plant breeding in a sequence. First emasculation is done. First emasculation is done. First selection of parents, then selfing in parents, then emasculation. After emasculation, it is bagging. After emasculation, it is bagging. Emasculation to avoid unwanted self-pollination. Bagging is to avoid unwanted cross-pollination. After bagging, artificial cross-pollination. The bags are removed, pollen grains are collected from male plant and deposited on the stigmas of the flowers. Again, bagging is done. So, bagging is done before pollination, after pollination. Then trials, multiplications and distribution to the farmers. So, the correct sequence is first emasculation, then bagging, then artificial cross pollination, finally trials. So, 3 emasculation first, 1. Bagging next. 2. Artificial cross pollination next. Final trials, multiplications and distribution to the farmers. So, 3, 1, 2, 4. Write the correct sequence of development of crop that can be reproduced sexually and vegetatively. Sexually and vegetatively. Mass selection is the first, where importance is given to cross pollinated crops. Then pure line selection is the method where importance is given to self pollinated crops. Self pollinated crops. Then hybridization is done for various cross and self pollinated crops. Finally, clonal selection, which is done only for vegetative crops. So, reproducing sexually and vegetatively. Sexually means cross pollination for mass selection, second option. Self pollination for pure line selection, first option. Hybridization in between two plants with different genotypes, pollination is not given that importance. And finally, vegetative propagation, clonal selection. What is a clone? A group of plants obtained from a single plant just by vegetative propagation is called as clone. So, 2, 1, 3, 4, fourth option. Write the correct sequence of development of breadwheat. First, formation of amphihaploid. First, formation of amphihaploid, that is F1 generation. Then formation of allotetraploid. When the F1 generation chromosomes are doubled, allotetraploid is formed. Then formation of allotriploid. Then allotriploid is formed, and formation of amphitriploid. How it is done? When Triticum monococcum 2n is equal to 14 is crossed with Aegilops speltoides. F1 generation is formed, chromosomal number is doubled, amphidiploid is formed. Amphidiploid is crossed with Aegilops squarosa. Aegilops squarosa. Now, triploid plant is formed. When chromosomal number is doubled, hexaploid plant is formed. Because this has 2n is equal to 14, this is 2n is equal to 14, this is 2n is equal to 14, here 4n is equal to 28, here 2n is equal to 14, here 3n is equal to 21, here 6n is equal to 42. So, this is the way how Triticum monococcum crossed with Aegilops speltoides. F1 generation is formed with 2n is equal to 24, 2n is equal to 14, doubling of chromosome takes place, amphidiploid is formed, it is crossed with Aegilops squarosa, and once Aegilops squarosa is crossed, triploid plant is produced. So, answer is 4. 3. Formation of amphihaploid F1 generation. 4. Formation of allotetraploid, chromosomal number is doubled. Second, formation of allotriploid, 3n variety is produced. Last, formation of amphitriploid. Amphitriploid is nothing but hexaploid variety of wheat. So, Triticum aestivum is produced by different main method, that is Triticum monococcum crossed with Aegilops speltoides. F1 generation is formed, chromosomal number is doubled, tetraploid plant is formed, it is crossed with Aegilops squarosa. A triploid variety is formed and doubling of chromosomes, hexaploid variety will come. So, 3, 4, 2, 1. Plant breeding, merit and demerit. Introduction, easiest method, choice of entry of pathogens. So, introduction is the easiest method, just transferring plants from one area to other area, one district to other district, one state to other state, one country to other country, one continent to other continent. But the demerit is there is a chance of entry of pathogens. Definitely, there is a pathogen called Phytophthora infestans is transported along with the potato introduction, which cause devastating effect like Irish famine, late blight of potato. Such pathogens may enter. Then hybridization don't require skill, expensive method. Hybridization requires maximum skill is required. So, second is wrong. Pure line selection applicable to self pollinated crops, very poor acclimatization. Pure line selection, definitely it is done only in self pollinated crops. It shows very poor acclimatization because they are phenotypically, genotypically similar. Very poor acclimatization is a bad demerit in all pure lines. Mutation breeding, always desirable mutations are obtained. A few plants show mutations. Always desirable mutations obtained we cannot say, and a few plants show mutations is wrong. Large population of mutations is correct. So many new plants has to be tested and very much carefully they are tested. So 1 and 3 are correct. What are they? Introduction, easiest method, chance of entry of pathogens. Pure line selection, applicable to self-pollinated crops, poor acclimatization. Hybridization don't require skill is wrong. It requires maximum skill. Mutation breeding, always desirable mutations are obtained is wrong. So 1 and 3 is correct. Now, variety, crop and plant breeding method. Texas 21, maize introduction correct. CO16, sugarcane is wrong. Rice. CO16, rice pure line selection is correct. Here sugarcane is wrong. Kufri red potato mass selection is wrong. Kufri red is potato correct, but not by mass selection, it is by clonal selection. Puzamoti bajra mass selection is correct. So Texas 21 variety of maize is obtained by introduction is correct. CO16 sugarcane is wrong. It is CO16 sugarcane is wrong. CO16 sugarcane is wrong. It is rice. Pure line selection is correct. Kufri red potato variety by mass selection is wrong. Kufri red potato variety is obtained by clonal selection. Puzamoti bajra by mass selection is correct. So answer is second option, first and 4. This is another 3 column matching. Column 1, column 2, column 3. Heterosis, Shull, tobacco. Pure line selection, Johansen, Phaseolus. Artificial cross pollination, Babylonians, Coccus. Mutation in plant breeding, Stadler, barley. Heterosis by Shull but not in tobacco, it is in maize. Heterosis, superiority of progeny over their parents is called heterosis. It is discovered by Shull only but not in tobacco, it is in maize plants. Pure line selection is discovered by W.L. Johansen in Phaseolus plants. Phaseolus vulgaris is correct. Phaseolus vulgaris is correct. Artificial cross pollination is Babylonians, Coccus is wrong. Artificial cross pollination is Babylonians but not in Coccus. Mutation in plant breeding. Mutations in plants for breeding. Stadler correct, barley is correct. Muller in Drosophila, Stadler in barley plants. So heterosis, GH Shull correct but not in tobacco plants. Pure line selection, Johansen is correct, Phaseolus vulgaris is correct. Then artificial cross pollination, Babylonians, Coccus is wrong. Mutations in plants for breeding, Stadler, barley is correct. So 2 and 4 are correct. Hybrid variety, male parent, female parent. Raphanobrassica, Raphanus brassica. Raphanus sativus and brassica is wrong. Raphanus is female parent, brassica is male parent. Pusaropi, Miruthi and Siox is correct. Triticum aestum and Mevigelops and Aegilops is the male parent and Triticum monococcum is the female parent, correct. White cotton, Gossypium hirsutum, Gossypium barbadens is wrong. So answer is 2 and 3, second option. This is another 3 column tabular form. Plant breeding method, variety, crop. Polyploidy breeding, TV29 coffee. TV29 coffee wrong. Polyploidy breeding, only wheat variety is given. Mutation breeding, Jagannath variety, rice, not barley, it is rice. Hybridization, red plum variety in tomato is correct. Pure line selection, HG18 variety of groundnut is correct. So 3 and 4 are correct, third option. Cotton variety, male parent, female parent. White cotton, Gossypium hirsutum, Gossypium barbadens correct. Devi Raj variety, Gossypium arboreum, Gossypium hirsutum is correct. Devi Tej and Dodahatti local is wrong. Dodahatti local is obtained by mass selection, not by hybridization. So 1 and 2 are correct. Arrange the following stages of hybridization in a sequence. Selection of parents, removal of anthers, keeping the pollen of male parent on stigmas, multiplication of the seed. First is selection of parents. First is always selection of parents. Then emasculation after selfing, inbreeding, after conducting inbreeding selfing, removal of anthers from a flower before anthesis, before the opening of flowers is correct. So removal of anthers to avoid unwanted self-pollination. Then 3. Keeping the pollen of male parent on stigmas is correct. Bagging is done, bag is removed, pollen grains are taken from male parent and deposited on the stigma of the female parental plant. So, keeping the pollen of male parent on stigma is correct. Then multiplication of seed is correct. So, all the 3 are 4 in correct sequence. Selection of parents, removal of anthers by emasculation, keeping the pollen of male parent on stigmas, artificial cross pollination, multiplication of seed. F1 generation is formed, seed is multiplied, F2 generation is produced, trials, multiplications and other aspects are taken into consideration. This is an assertion. Progeny obtained by pure line selection are phenotypically and genotypically uniform. Definitely progeny produced by pure line selection are phenotypically and genotypically uniform. Why? Pure line selection results in homozygosity in progeny. So, pure line selection, what is a pure line? Pure line is the progeny of a single self-fertilized homozygous plant. Single self-fertilized homozygous plant is pure line. So, pure line selection always results in homozygosity, increases homozygosity in the plants. So, both A and R are correct. R is the correct explanation of A. Match the following. Cold treatment to zygote when it is in dividing stage you can produce in polyploidy breeding 4. Incorporation of several desirable characters into a single variety is done. Incorporation of several new varieties into a single variety is done by emasculation. Exploiting hybrid vigor for many generations is done in clonal selection. Hybrid vigor can be taken for many generations. Improving local varieties of self-pollinated crops is pure line selection. So, cold treatment to zygote when it is in dividing stage is polyploidy breeding. Incorporation of several desirable characters into a single variety is done by hybridization. And also this one, and exploitation of hybrid vigor is by clonal selection, and improving local varieties of self-pollination varieties is done by pure line selection. A for B3, C1, D2, fourth option. Emasculation. It is a prerequisite in crossing the normal self-pollinated crop. Definitely normal self-pollinated crop stamens must be removed. Need not be done in the flowers of male parental plant. It is not done in male parental plant. Need not be done in male sterile flowers. Correct. Not applicable to dioecious plants. So, 1, 2, 3, 4 are correct regarding emasculation. During hybridization, bagging is done. During hybridization, bagging is done. What is bagging? After emasculation, removal of stamens from the bisexual flowers of female parental plant. To avoid unwanted self-pollination, bagging is done to avoid unwanted cross-pollination. So, before pollination, immediately after emasculation, it is done. Then bagging, the removal of bags takes place. Pollen grains are collected from male parental plant, deposited on the stigmas. Again bagging is done. So, after emasculation only is wrong. Before and after emasculation is wrong. Before artificial cross-pollination only is wrong. Both after emasculation and artificial cross-pollination. Why after emasculation? To avoid unwanted self-pollination. Why after cross-pollination? To avoid unwanted cross-pollination. So, both after emasculation and artificial cross-pollination, during hybridization, bagging is definitely done. Third option. In vegetatively propagated heterozygous crops, the heterozygosity remains same. The breeding due to the fact that these crops do not produce seeds at all is wrong. Selection is based on mutation is wrong. All the progeny of a clone is similar in phenotype is wrong. Vegetative propagation does not involve segregation of character is correct. Because the vegetative propagation does not involve the segregation of characters, in vegetatively propagated crops, heterozygous crops, particularly in vegetatively propagated heterozygous crops, the heterozygosity remains the same the breeding due to fact that in vegetative propagation there is no involvement of segregation of characters. So, third option. Regarding pure line selection, it gives uniform progeny. Uniform progeny means they are phenotypically and genotypically similar. Progeny shows same phenotype and genotype. So uniform progeny is correct. Progeny shows phenotype and genotype similar. Increases the homozygosity of the progeny. Pure line is nothing but self-pollination in a self-pollinated crop, that is called as pure line selection or pure line. So increases homozygosity of the progeny is correct. RSB 17 in groundnut and CO4, 6, 10, 14 varieties of rice are developed through pure line selection. So given uniform progeny is correct. Progeny shows same type of phenotype and genotype is correct. Increases the homozygosity of the progeny is correct. RSB 17 in groundnut and CO4 in rice are varieties developed through pure line selection is correct. So regarding pure line selection, 1, 2, 3, 4 are correct. Assertion. Colchicine acts as both chemical mutagen and as a chemical to produce polyploids. Colchicine is obtained from the seeds and culms of a liliaceae plant called Colchicum autumnale. Colchicine is widely used as a chemical mutagen. How? It prevents the formation of spindle fibers during metaphase. When spindle fibers are not formed, the chromosomes are not separated and they get doubled. So they help in the formation of polyploids. So colchicine acts as both chemical mutagen and chemical to produce polyploids. It is basically chemical mutagen and a chemical to produce polyploids. Correct. Colchicine is an alkaloid. Because colchicine is an alkaloid, correct. But because colchicine is an alkaloid, it acts as both chemical mutagen and chemical to produce polyploids is not that related. So both are correct, but reason is not the correct explanation. So answer will be both A and R are correct. R is not the correct explanation of A. So colchicine acts both as chemical mutagen and chemical to produce polyploids is correct. Colchicine is an alkaloid, correct, but not because it is alkaloid, it acts as both chemical mutagen and chemical to produce polyploids. So second option. Next. The progeny obtained by pure line selection are phenotypically, genotypically similar. Pure line selection increases homozygosity. Because pure line selection increases homozygosity only, the progeny obtained by pure line selection are phenotypically and genotypically similar. So pure lines always useful to improve local self-pollinated crops. Pure lines show all homozygous genes. Inbred lines show. Inbreeding means conduction of self-pollination in self-pollinated crops is pure line. Conducting self-pollination in cross-pollinated crops is inbreeding, and the resultant ones are inbred lines. Pure lines contain all homozygous genes. Inbred lines contain some homozygous and some heterozygous genes. So the progeny obtained by pure line selection are phenotypically, genotypically similar is correct. Pure line selection increases homozygosity. Because pure line selection increases homozygosity, the progeny obtained by pure line selection are phenotypically, genotypically similar. So both A and R are correct. R is the correct explanation of A. Now, arrange the following stages of hybridization process in ascending sequence. Ascending sequence means first to last. First is selection of parents. After the selection of parents, selfing in parents is conducted. Selfing in parents is not given, emasculation. So 2, 4. Then after emasculation, it is bagging, 3. Then after bagging, it is artificial cross pollination, removing bags and depositing pollen grains on the stigmas. And finally, self pollination to produce F1 generation. So 2. Selection of parents, 4. Emasculation, 3. Bagging, 5. Artificial cross pollination, last self pollination in F1 generation. So always first step is selection of parents. Importance is given to ecotypes. Ecotypes means the plants which are selected by nature. Importance is given to ecotypes. Then self-pollination is conducted even in cross-pollinated crops to improve homozygosity and reduce heterozygosity. Then emasculation, removal of stamens. Then bagging. Then artificial cross-pollination. Then growing F1 generation. Then by self-crossing or intercrossing F1 plants, F2 plants are produced. Then F2 plant seeds are collected. They are taken for trials in various research stations. Once they prove to be very good, they are allowed for multiplication, and finally they are distributed to farmers as the best varieties. So selection of parents, emasculation, bagging, artificial cross-pollination, self-pollination in F1 generation. The process of segregation of genomes will not occur in the progeny obtained by. Segregation of genomes means separation of genomes entering the other plants, that means crossing and all. In polyploidy breeding, segregation of genomes will be there. Why? Because in polyploidy breeding, 2 plants or 3 plants or 2 plants are crossed and genes get segregated. Pure line selection is self-pollination. Definitely segregation of genomes will be there. Pure line selection is nothing but self-pollination. Whenever self-pollination, there is chance of segregation. Mass selection is cross-pollination. Definitely segregation of genes takes place. Clonal selection is vegetative propagation where segregation of genes absolutely not occur. So answer is 3 only. So the process of segregation of genes will not occur in the progeny obtained by mass selection. So these are some of the important bits regarding genetic code, protein synthesis, phytohormones and crop improvement. In the next class we discuss one class for biotechnology, genetic engineering, one class for tissue culture, single cell protein and mushroom cultivation.